Have you ever wondered, why do we age? What tells our bodies to get older? Why does our skin wrinkle, our hair gray, and our immune system weaken? What tells our body, your time is up? Perhaps you have even wondered, could we live forever? Cue the telomere. Telomeres are repetitive nucleotide sequences located at the termini of each of our chromosomes. They act sort of like an aglet, the little plastic tip at the end of a shoelace. Telomeres keep chromosome ends from fraying, sticking together, and getting damaged. When a cell divides, its chromosomes are replicated so that each daughter cell receives genetic material identical to that of the parent cell. In this process, an enzyme called a helicase unzips the parent strand. A DNA polymerase, a nucleotide-producing machine directed by a primer sequence of RNA, then synthesizes base pairs complementary to the leading and lagging strands of DNA. However, each resulting strand is a few base pairs shorter than the original, as some room is taken up by the RNA primer sequence responsible for activating the DNA polymerase. Because of this, each time a cell divides, the telomeres get a little bit shorter, 50 to 250 base pairs per division in human cells. Why is this important? Without telomeres, the shortening of chromosome sequences would still occur each time a cell replicated. However, this time, the removed base pairs would come from essential genes housed within the body of the chromosome, altering or even preventing their function. Okay, so now we know what telomeres are, how they function, and why we need them. Now, let's look at what they actually do. Telomeres are the cell's internal clock. They tell the cell how much longer to live and when to die. This is because, as telomeres shorten with each division, they eventually reach a critical length. At this critical length, the cell division process becomes less active and the cells eventually cease to divide. Here's where things get interesting. In germline cells, egg and sperm cells, there's a nifty little enzyme called telomerase. Telomerase functions to counteract the telomere shortening that happens every time the cell divides by adding nucleotides to the end of the resulting strand. Without this shortening of telomeres, a cell's division process is able to continue indefinitely. In fact, this phenomenon is not unique to just germline cells, but is also a distinct trait in the proliferative success of cancer cells. In order for cancer cells to continue their rapid and unending process of replication, they employ the telomerase enzyme. In its absence, cancer cells would be unable to propagate indefinitely. This concept begs a question that has been circulating in scientific literature since the 1980s. If telomerase can make cancer cells and germline cells effectively immortal, could we use telomerases to make all cells immortal? In the past, the roles of telomeres and telomerases have been undercharacterized in experimental literature due to an inability to affect real-time change within living cells. However, thanks to the rise of precise and efficient genetic editing mechanisms such as CRISPR-Cas9, we are now at a place where in vivo studies of these cell components are within reach. As of 2018, the average life expectancy of an American was about 79 years old. If we could find a way to immortalize all dividing cells via telomere lengthening in a non-destructive way, what could that number be?